This is the workshop on importing data with rich metadata at scale. Following on from Colin's workshop describing command line tools for importing data, we're going to talk about the specific pipeline we have used for publishing data in the image data repository. So Josh and myself are going to give you a short introduction describing the pipeline that we've developed. Sorry, I'm just trying to find where to move down the page. Here we are. So first of all, Josh will give an outline of our metadata pipeline that we use for the image data repository and when it's suitable for use. And then I'll talk about what kinds of metadata we're adding, how do we record the metadata. Then we'll be back to Josh and he'll talk about how we load it into OMERO, and then we'll both talk about what we're thinking about next. So over to Josh to tell you about the pipeline.
And so most importantly, it's to keep in mind that this is more of a typical publication workflow. So we're getting all the data in large batches up front from the author. And then after some communication with the author, we can upload it independently. There's no ongoing analysis or further changes to the data once we get it in our hands. So what that allows us to do is take batches that work for us, and these go from anywhere from, so a single import is from, say, 1 hour to 12 hours, and then put them together in bunches of up to 500 or maybe 1,000 imports and let it just run for long periods of time. And then after the import, we run a second phase, which is generating rendering settings, and then after that, we can attach the annotations. So we really have a lot of autonomy once we've begun working. That's not feasible in all cases, but here it's worked out quite nicely. Eleanor?
Yep. So we thought it might be useful if we describe two different data sets that we've received for the IDR project and tell you about the experimental and analytic metadata that we imported for each. So the first is a super resolution study of the nuclear pore complex. They looked at 7 proteins in the complex, and this produced around 500 image stacks. They used different antibody techniques to visualize the proteins and try to work out their position within the nuclear pore. 500 images are not a vast number, but it's still more than you would like to annotate by hand. So I'm going to show you what metadata we added to those images and how we imported it. The second dataset was much larger. It's the Metadata dataset, which is a high content screen looking at the knockdown of around all the human genes, 22,000 human genes over 500 plates, and there was around 200,000 wells. In this, they were looking at time-lapse of dividing chromosomes and did both manual and computational analysis of phenotypes. So in each case, we tried to pull out information from the files that were sent, and I've listed some of the examples here. In the nuclear pore complex case, I got most of the information from the publication. In the case of Metadata, they provided text files with some results, but most of their experimental metadata about siRNAs and their sequences and the genes they were targeting was in a MySQL database, so they provided me a dump of that. So we tried to organize and standardize the information from both data sets and put it into standardized columns of data, and the things that I've highlighted in orange are things that I've added to the data sets. So for example, in the nuclear pore complex data set, they gave me the protein name, but so that we could link this to other data sets, I added also what the corresponding gene identifier would be and the gene symbol. And in both cases, we added some phenotype ontology annotations. So here's an example of me adding the gene identifier to the nuclear pore dataset. So here we have the image, the targeted protein name, and here I've got the gene identifier and the gene symbol and some gene symbol synonyms. Here's an example of what it looks like when I add the phenotype ontology mapping. So this is from Mitacheck. So one of their phenotypes was nuclei stay close together. We mapped that to binuclear cell phenotype and Campo, and here's the Campo accession number. So in each case, we put the information into a set of three standard files: the study file, the library or assay file, depending on whether it was a high content screen or not, and the results file. So the study file includes things like the study type, the imaging method, descriptions, and also I put the phenotype ontology mappings in there. The library or assay file has reagent identifiers, so things like the siRNAs or compounds, if it's a compound screen, gene identifiers, information about which were control wells, and any quality control information, so which wells were rejected because there was, for example, too few cells. Then in the results file, we have the measurements, the results of the image analysis, any measurements they made, maybe counts of cells, scores they've made on time spent in different phases of mitosis, for example, and the phenotypes that they observed if they recorded this. And then what we do is we pull out different bits of that information and put it into an annotation CSV file, so a comma-separated file, and this is what we can load into the OMERO database. If you'd like to see examples of the study library, assay files, or results files, and also of the annotation CSV files, you can find them in GitHub at this location, as listed down here. Here I've made a made-up example of the annotation CSV file, because otherwise the values are too long, but just to show you what one looks like and where the different bits of information come from. So this is a high content screen example, so you can see that I've got the plate and well listed, what siRNA was the cells were treated with in each well, the genes that it was targeting and the gene symbol, any information about controls. And then this is from the library file. And then from the results file, we have a score that they've determined, and then some phenotype information. And then I've pulled from where I've recorded in the study file what the phenotype to ontology mappings are. And I have a Perl script which pulls this information together. So what does the data look like when we load these CSV files into OMERO? Well, it's loaded as a bulk annotation file, and here you can see one of the tables with all different bits of information. This is from the Mitacheck screen, so you can see the siRNA identifier. This is actually a negative control, so there was no target gene, but you can see the sequence of the siRNA, and then information about scores from the cells in that well. And you can go and look at this data yourself by going to this URL down here, idrdemo.openmicroscopy.org. And then I'll pass you back to Josh, who's going to tell you a bit more about how we actually go about loading the CSV files into OMERO.
Thanks, Eleanor. So the CSV that Eleanor generated with Perl, so this annotation CSV, is something that's been importable to OMERO, at least for many versions. I don't even remember how long. So it generates what's called a bulk annotation, or also known as an OMERO table. So that's stored on the file system as a HDF5. The script to do this is available, and anyone can prepare a CSV file. The only thing that's required is that there's at least one column which specifies where the annotation should get attached in OMERO. So Eleanor showed a plate column, and so that said this is going to be attached to a screen, and the plate names have to match. So each plate in the screen matches one of the plates in the CSV file. And this works fine, has been available for some time. Unfortunately, since it's an external file, it's not in the Postgres database, there's limited functionality when trying to query it. And we realized that there's also the problem that in the UI, so in the previous screenshot, there are just a number of fields that show up in the right-hand panel that you don't really need. So all the float values become just too much. So if you have hundreds and hundreds of columns in your CSV, it's not helping you define what you're looking for. So what we decided to do is to take the most critical columns from the OMERO table and turn them into map annotations. So that's what we're talking about here. There's a configuration file written in YAML which selects a number of columns and possibly maps them. So if your column names in your CSV are perhaps a bit cryptic, you can give them nice or more user-friendly names. And so here you can see we've taken the gene identifier, and it's available just as a string, but then we've also turned that into a URL so that the user can click on the identifier and go to the Ensemble webpage. The other big benefit of this is that since it's in Postgres, it gets automatically indexed into Lucene, so that means all the values that you put in the map annotations are immediately searchable. Go to the next slide. So so that's an example. You have the gene identifier, and this is exactly what you would put into the mapping file, into the mapping YAML, so that you can specify how you want them displayed, and perhaps in some examples you can hide you can turn some of the mappings on and off as you need. And then the next slide, and this is driven by this OMERO metadata populate command. And as I mentioned, as long as you set up the CSV correctly, you have the appropriate columns, you can attach these kind of annotations to images at the screen, the plate, the project, or the dataset level. So depending on what level of granularity you've ordered your CSVs into, so you can have one very large CSV and you put all the information in it, or you can break it down and have, say, one per plate or one per data set. And this is generally a process that we've found to work quite well, that we've taken what perhaps has been a manual process, put it into a configuration file, and that's what's helped us to build up what we're calling the data pipeline of getting the entire publication in over a long period of time. And then we've taken that and applied it to other bits of the process. So Colin was talking about imports, and there are a number of command line arguments you can use to get the data in, but you can only do that so many times before it just becomes limiting. And so what we've done is we've taken all those configuration properties, again, put them into a YAML configuration file, and that lets you specify everything up front once, and just rerun the imports as necessary. So these configuration files are in the same GitHub repository that Eleanor spoke of. So you can see, really exactly what we do from beginning to end to have the IDR data imported. And the next slide. Same thing for setting the rendering settings. So usually each screen has a single rendering setting that makes sense for all of the images. That helps you kind of detect the outliers. And rather than trying to drive that via the UI, there's the functionality in OMERO and the GUIs that you can find an image and you can set the rendering setting, and then you can apply that rendering setting across the entire plate. We've kept in our same process, defined a YAML file. All the information goes into it. It's checked into GitHub, and then you can apply it as necessary. And then we would actually like to do the same thing with regions of interest. So far, we haven't found enough commonality between the various screen submissions to say, okay, here's how you specify where regions of interest are stored, but we're investigating that. And that's the beginning of our to-do list, so that's what we'll cover next.
Yes, so one of the things we want to do is to improve our display of search results. For the moment, if you search for the gene, for example, pipox, you get a list of whales back, but you can't see exactly which study they come from. So we'd like to include this information in that view. When you click on a whale, it one of these thumbnails, it takes you to the whole screen, and it's quite hard to realize which well you originally were looking for. So that again is something we want to improve on. And also we want to give you more options in the search. So at the minute, if you search, this is in the top search box on the OMERO screen. If you search for something, it searches across all data sets and screens. And what we'd like to be able to do is say, I want to search just within a particular screen or data set. We'd also like to make more use of ontologies. So we have most of the phenotypes observed are mapped to an ontology term using the Campo ontology, the cellular microscopy phenotype ontology. And we want to make use of the ontology hierarchy so that you can browse more easily the phenotypes. And the ontology lookup service at EBI provides some widgets for doing this, so we'd like to incorporate them into OMERO. And we're also looking at things like bulk annotation of ROIs and using folders. And so you might have genes, your folders by gene, so you can then look at all ROIs from different images that are related to a particular gene. And we might do this for phenotypes as well. Then we're ongoing work on standardizing the templates. So we've made an initial start based on work of others in the cellular phenotype database and drawing from ISATAB and MageTab experiences. But we need to carry on looking at other types of data sets, so light sheet microscopy, plant studies, medical imaging, see how these would work in with what we've already created, and then also align our efforts with other projects which are also looking at standardization, such as the multi-MUP project. And then back over to Josh.
And most important, I guess, from the side of running this pipeline and getting the data in is that we would really like to make it non-manual. So we've recorded as much as we can to make it reproducible, but there's still a lot of human intervention. What would be ideal is then to really take the entire package, so all the YAML files, all the image files, all the metadata files, and really just give them to the server and have them all imported in one go. That's feasible, but especially when dealing with this many individual steps, there is a failure rate, and it becomes tricky to detect what's gone wrong. So hand in hand is the idea of having the server detect any errors and somehow feed them back to the user. Is that by sending an email? What happens though if 1200 failed and you get 1200 emails? Is that a web page that the user has to go to? So there needs to be some system of back and forth between submitter and server so that all of this can happen in a more automatic fashion.
So finally, just to finish up, here's the URL again for the IDR prototype. So idr-demo.openmicroscopy.org. So if you want to have a look at the screens and other data sets that we've got in, you can go here. And also, we just like to thank all the team in the OME group in Dundee and the group in Cambridge and EBI, and also Glencoe Software for all their help on this project.